Welcome to This Explainer. Today, we're diving into the absolute gauntlet that new therapies have to run to get from a lab bench directly to a patient. If you've ever wondered about the incredibly rigorous, step-by-step pathway a new medicine has to take before it finally lands on a pharmacy shelf, well, you're in exactly the right place. We're going to break down this complex timeline into a really clear, logical journey.
Here is our roadmap for today. We'll start with the basics, move into the preclinical phase, tackle phases 1 through 4, touch on study designs, and then wrap it all up with a quick summary. Let's get into it.
All right, Section 1, the basics of clinical research.
So, what exactly is a clinical trial? At its core, it's a highly structured scientific investigation designed to test both the safety and the efficacy of different therapies in human subjects. You can really boil this entire concept down to just 3 key words. First, you have the experimental unit. That's the actual subject participating in the trial. Second, the treatment, which is the specific intervention being tested. And third, the evaluation. This is where we rigorously measure the outcomes. Does it work and is it safe? Now, depending on the specific medical goal, researchers categorize these trials into distinct types. It's actually not always about curing someone who is currently sick. For instance, while treatment trials do test new drugs or surgeries, prevention trials look for ways to prevent a disease in people who have never even had it, or stop it from coming back. We've also got diagnostic trials to find better testing procedures, screening trials for early detection, and quality of life trials, sometimes called supportive care trials, which are all about finding ways to improve comfort for folks living with chronic illnesses.
Moving right along to Section 2, the preclinical phase.
Think of this timeline like a series of heavily guarded checkpoints, escalating all the way from the lab up to global post-approval surveillance. A drug simply cannot pass to the next stage without fully clearing the rigorous requirements of the one right before it. Each phase is treated as an entirely separate clinical trial.
Before pharmaceutical companies even think about starting trials on human subjects, they have to put the drug through extensive preclinical testing. We're talking in vitro experiments, so in a test tube or a cell culture, and in vivo experiments, which use animal models. Researchers will test wildly varying doses here. The whole point of this hurdle is to gather preliminary efficacy, toxicity, and pharmacokinetic data. Basically, they need to figure out if this candidate has enough real scientific merit to even be considered an investigational new drug, or IND.
Section 3, we are officially entering human testing with Phase 1.
Phase I is the big debut. These are the absolute earliest trials in the life of a new treatment. They're usually very small, maybe up to 30 people. And here's a key detail. These are mainly healthy volunteers, closely monitored in an inpatient clinic by full-time staff. At this point, the primary mission isn't necessarily curing a disease. It's purely focused on safety and tolerability. They're looking for significant adverse events and trying to establish a safe dose range. They're also intensely studying pharmacokinetics, how the drug is absorbed and eliminated, and pharmacodynamics, which is how the drug actually affects the body.
Next up, Section 4, Phase 2, clinical trials.
Once safety is established, we push into Phase 2, and the stakes definitely go up. Researchers recruit a larger group, typically between 20 and 300 volunteers. But there's a huge shift here. These are now actual patients who have the specific disease being studied. It's a highly homogeneous, carefully selected group. While they're still monitoring safety and short-term side effects, the focus pivots hard to therapeutic exploration. They want to know, does this drug actually work for its intended purpose? And crucially, what exact doses should be used in the next massive phase?
Now, in the real world, Phase 2 usually gets split into 2 parts. You've got Phase 2A, which is specifically designed to figure out dosing requirements. Literally, just answering, how much of this drug should we give? Then, Phase 2B takes those prescribed doses and zeroes in on efficacy, determining exactly how well the drug works at that specific level. It's an incredibly methodical stepping stone.
All right, Section 5, Phase 3 trials. This is the big one, Phase 3. We are talking massive, randomized, controlled, multi-center trials. Depending on the condition, you might have anywhere from 300 to well over 3,000 patients. The goal here is definitive assessment. We aren't just asking, does it work anymore? We are asking, is it actually better than what we already use? The new drug is rigorously compared against current gold standard treatments. If it proves satisfactory, all of that data, the human trials, the animal studies, the manufacturing procedures, gets bundled into a massive regulatory submission. If the national authorities approve that document, the sponsor can finally market the drug.
Which brings us to Section 6, Phase 4.
So, the drug is approved and out in the real world. We're done, right? Actually, scratch that. Evaluation pushes right into Phase 4, also known as post-marketing surveillance. This is absolutely crucial because early trial environments are limited in time and scope. Phase 4 involves pharmacovigilance. This is where researchers catch those super rare, 1 in a million adverse reactions. It's also where they track chronic toxicity over years of use and observe how the drug affects special groups, like kids or pregnant women, who generally aren't included in earlier, stricter trials.
Let's pivot quickly to Section 7, Study Designs.
How are these trials actually built? Researchers basically use two broad blueprints to gather their data: observational designs and experimental designs. Within those two broad categories, depending on the phase and the specific data they need, they'll use precise methodologies. Things like case reports, case series, and cross-sectional studies. These provide the statistical tools required to accurately evaluate their findings.
Finally, Section 8. Let's summarize the whole shebang.
Take a look at how this all comes together. Notice how the sample sizes just exponentially explode, from maybe 30 individuals in Phase 1 to thousands in Phase 3, and eventually to the entire general population in Phase 4. You can also see the participant shift, starting with mainly healthy volunteers, moving to very specific patient groups, and then expanding outward. But most importantly, track the purpose. It scales from basic human safety to proving efficacy to confirming a global therapeutic benefit, and finally, to long-term surveillance.
Knowing all of this, consider how this rigorous step-by-step pipeline guarantees that a new medication doesn't just magically appear. It is systematically tested, endlessly evaluated, and relentlessly vetted. So how does understanding this absolute gauntlet change your perspective on the medical breakthroughs of tomorrow? For sure, it highlights an incredible scientific architecture designed to ensure that only the safest, most proven therapies actually reach the patients who need them. Thanks for joining this explainer, and keep exploring the science behind the data.